I guess is getting out to the stuff like this and hearing some of the issues is really pushes us in our directions for research and trying to manage some of this stuff. So I've been given a pretty big brief here, so every disease in every winter crop, I think Nick said, so we might narrow it down as we go along. So 2015, so it's actually a joint thing. I work on winter cereals, so wheat, barley, oats to a lesser extent, triticale. So that's the crop I work on. So in terms of canola and the pulses, I've went on Kurt Lindbergh and a Wagga to present some information there which he had. So disease managed 2015, or as I like to call it, information overload. But hopefully through this process I can teach you, I guess the way I think about some of these diseases, it's about understanding how they work, I think then drives your management decisions and some of those timings and what we're doing. Going to quickly cover rust, okay? Anyone had rust problems the last couple of years, or it's dropped off the radar? Fungicide's pretty effective, generally? Okay. The reason I want to cover rust is we're going to then go into yellow spot. And what I want to draw out is the big difference between the rust and something like the net blotches and yellow spot, okay? With rust we're talking about a biotroph. You don't need to remember that. All that means is that it needs a living cell to survive. Okay, so it doesn't kill the cells, it parasitises them and lives inside the cells, and that's how it gets its nutrients. Okay, we've got different species and different diseases. We've got leaf rust, stem rust, which fortunately we've got very good resistance, so we don't have an issue with. If we keep those varieties out of the north, okay, they have issues in South Australia because they're growing susceptible varieties. We don't want them up here. If this ever got up into this region and further north, this would be our worst disease. There's probably some older blokes here might remember stem rust. This will kill a crop. Then we've got yellow rust, which is called stripe rust. Yellow rust everywhere else, but stripe rust here, okay? What they're doing, with the exception of this guy, which can take out the stem, is they're getting rid of green leaf area. They remove green leaf area. So you don't get the photosynthesis. That's how it's knocking yield, knocking screenings. Okay, so what we're trying to do is manage diseases like this to maintain that green leaf area, which gives us the yield. But what it comes down to is which leaves are actually important for driving that yield, which in wheat, it's the top three. Okay, biotrophs, they need that living plant. Who's heard of adult plant resistance around rust? Probably one of the most misunderstood things ever, and when it kicks in. So adult plant resistance is a mechanism that's in certain varieties that when it senses it's got the stripe rust fungus in it, it kills those cells. So the plant, it's deliberate cell death. It kills those cells and it kills the surrounding cells. So being a biotroph, it's no longer got living cells to feed on. So you kill the infection. Okay, so that's how adult plant resistance is. The vagary comes in around when that expression actually comes in. Varies with variety. Some of the genes come in from that second node onwards, and some don't come until full head emergence. And the big problem, which we'll go into suntop too here, many people growing suntop, becoming a more... We've had issues with stripe rust and suntop around nitrogen management, because the actual nitrogen management can delay expression of the APR genes in some varieties. And pretty easy to identify, hopefully everyone's up on that. Okay, last year generally, fungicides, most people putting a fungicide in with their broadleaf spray these days. Very common as you go further north. That is keeping the pressure out of the system, okay? But that's going on pretty much every variety, which probably isn't warranted. Certainly stripe rust and leaf rust around. The point I want to make here is they haven't gone away. It's actually management which is keeping this at low levels. We had unsprayed national variety trials, Spring Ridge, Tamworth, developed quite high levels of stripe rust. So we go back to 2013, there was a fair bit of hype around stripe rust levels, unexpected stripe rust levels in Spitfire. Anyone get involved in that? Here, press? Okay. I was quite concerned, so I guess probably 90% of the samples I saw in 2013 were stripe rust on Spitfire. What ended up coming back to is off-types in the Spitfire. So there was off-types which were more susceptible to stripe rust, so they're taking quite a high level, and you're getting these scattered plants in a crop which were getting stripe rust. Okay, and you can see that in some of the head types in Spitfire too. Last year it was really replaced with reports of stripe rust in suntop. Suntop is a moderate resistant variety. We shouldn't be seeing stripe rust development in suntop. I'll go into that. Livingston growers? No? Livingston? Okay, we've got different pathotypes. And what that is, pathotype is just a type of the pathogen. It's changed and it's taken out particular genes. So the one that really caused the big problems when we had a mutation in 2006 that took out a gene, a yellow rust gene, stripe rust gene, YR17. So that's when your Crusader become quite susceptible, and a range of other varieties. That's mutated again in 2011, up and down south, and it's taken out the YR17 gene and also the YR27 gene. Okay, so this is what is in Livingston. Livingston's got YR17 and YR27. So when this guy was only around, it was just YR17. The 27 gene was protecting Livingston. STAT's come in and spread, so it's taken a couple of years to spread from the south up into here. It is established through this region now. So if you've got Livingston, you need to manage and monitor with Livingston. And I'll just quickly go through some leaf rust pathotypes, okay? It's not meant to be the Grim Reaper talk, just an awareness of what's developing around us and to keep an eye on. So what there is, anyone Naparoo? No Naparoo? No, this is purely a Naparoo issue. And we've got another mutation, or an exotic introduction actually, out of into South Australia, which we need to keep an eye on. But I predict this will not be an issue for us for the next couple of years. It usually takes a couple of years for it to spread from south and move its way up. Just to make the point, this is our trial at Tamworth unsprayed. This is Mace, which is 80% of the Western Australian crop, and it's about, well, somewhere around 60% of the South Australian crop. Okay, they have to manage it in South Australia because they have the YR17 pathotype. So it relies solely on the YR17 gene. The reason it's 80% of the crop in WA is they haven't had the mutation and they haven't got that YR17 pathotype. They've still got the original one that came in over there in 2001. Okay, so they don't have to spray for stripe rust in WA. If they get that mutation or if they get that over there, this is going to see Mace drop out pretty quick, I would have thought. And there's other good stuff coming through there. Pathotype in our trial at Tamworth was YR1727. One is where there's no pustules, just on a scale. Line is where the whole thing's yellow, so that picture of Mace. And you can see something like Livingston sitting here, you know, so losing around 40% of its leaf area to stripe rust if you don't manage it. Okay, so you've lost that photosynthetic area. The point I want to make here too is this is Suntop out of the NPT, very few pustules, very old pustules. This is actually two Suntop sources. So we had the problem with rust in Suntop around Wellington in 2013. We got hold of the seed from those crops, put it in here. We couldn't get any pustules on it at all, even though we know we get an infection. Got Mace getting the heat. Okay, this was a dryland trial with 50 units of N, so what's that about? Just under 100 kilo of urea. Okay, so not big N management, and that'll become important. Stripe rust and Suntop. Okay, Suntop's been in trials since 2010. These are dedicated stripe rust trials. It's always reacted moderate resistance, so it hasn't been getting pustules, except for one trial in 2013 where it did take a bit. Okay, so it's not like we've had a pathotype change. 2013 there was limited reports around Wellington where they're getting stripe rust in Suntop at concerning levels, and last year we're pretty much down south of here, up on the Liverpool Plains. There was reports of stripe rust in Suntop. Inevitably, the high rainfall areas, well, they were irrigated crops, and what they were trying to do is correct for the protein dilution of Suntop. Suntop Gregory, high yielding varieties don't know when to stop, so when they keep yielding under those conditions, you get protein dilution by it keeps putting carbohydrate on in the seed. Okay, so what they're trying to do to still get prime hard is put a heap of N under it, and sometimes split applications to up the protein, keep the protein levels up. We thought it might have been a pathotype issue, so Suntop has these three genes, YR1727, and it's also got an adult plant resistance gene 31. So there's a bit of a theory going on mid-season. Well, the YR1727 pathotypes become spread, therefore we're back to one gene, and that's why Suntop. But when you look at it, there was no evidence that that's what's going on. The majority of what was coming off Suntop was just the straight YR17. We know it's pure seeds. We tested the stuff from Wellington, we tested the stuff around Narromine, we had a problem as well. It was definitely Suntop. There wasn't off types in it, okay? And the pattern didn't indicate a contamination issue. If you've got contamination with a more susceptible variety, you'll see individual scattered plants with a lot more disease on them. The adjacent plants may get a little bit of spotting, but once you get away from that, the resistance keeps it out if it's the true variety. Okay, so the wash-up. Suntop, Spitfire varieties like that, they do rely quite heavily on that adult plant resistance. They can be susceptible to seedlings. We saw Gregory back in about 2008, a bit early on. These APR genes, they get delayed by high nitrogen stations. So the higher the nitrate level in the leaf delays the expression of some of those genes. Okay, and certainly when you've got irrigation or you're in a higher rainfall, it's more conducive, bigger biomass, and you drive the cycle. So certainly not going to be relevant to you guys. I don't think you're generally high rainfall. You wish, but if you are high rainfall and you are going to chuck a heap of N on, which probably from what I'm hearing neither are. But for these guys, the recommendation, if you're chasing high protein in these varieties and you go on irrigation, then you need to manage the rust. And it's only a one spray strategy in there at around that growth stage 30. And monitor. Okay, in terms of spraying, so if you're going to do an in-crop spray, which is the money spray? So this is some older work. So what I call high is where you've got high rainfall or irrigated and more conducive. We looked at a growth stage 32, so second node spray and a full flag leaf spray, versus just putting it in at that growth stage 32. Okay, so that's where the third leaf downs fully emerge. You can see in a high situation, your 32 spray is doing half the job. You get half your yield benefit from your 32 and the other half from protecting your flag leaf, which is your main photosynthetic area. When you get into your environment, we go west of the mule generally, not as conducive to rust, and crops tend to mature earlier, so that heading comes in quicker, less delay, less time between 32 and full head emergence. And what you find there is, and this is across numerous trials, 80% of your actual yield benefit from a fungicide is the 32 only spray. And you're chasing diminishing stuff with the second spray. Okay, so very different from when we first started talking stripe rust. We just say you've got to wait for the flag fully out, get us, protect the leaves. We wait for 39 in this environment, we lose too much of our mid-canopy in those susceptible varieties. And in most years, we get away with just that earlier spray. Do you need to spray all varieties? The answer is no. Skilgandra, it was only the YR17 pathotype, which is why Livingston looked right. Full control was up front, so flutriafol on starter fertiliser, a tebuconazole spray at 250 mils per hectare at 32 and another one at 39. So 3 fungicide inputs. We didn't get rust where we had full control. The only time we're getting yield loss, so we're getting significant disease development that actually takes out enough leaf area, is we get moderately susceptible varieties. So the key thing here, if you've got a moderate resistant variety or better, there's no benefit from fungicides. Okay, even the early ones. There is no cost to the plant to fighting rust. So a resistant variety, what happens, the spore will still land on the leaf, they germinate and they enter through the stomates, which are the gas exchange cells, and they'll infect 1 or 2 leaves, the plant takes it out. You won't see any loss of photosynthetic area, not perceptible. The big driver with this, or the big caveat on this, is you've got to make sure you've got the right variety, you're dealing with the right variety and it's pure. Okay, we go back to Canamble, I think it was, I can't remember what year it was now, I think it was 2010. Ran Canamble, a lot of Gregory got sprayed for stripe rust. Okay, a very good agronomist sent photos over saying, look at my Gregory, it's fallen over, it's full of stripe rust, and I sent him back a message saying, it doesn't look like Gregory to me, it's got a very fat flag leaf. It's just not what I'm used to in Gregory. That sort of went on for about a 3-week exchange. In the meantime, this went through the pubs, went through the whole district, and a heap of Gregory got sprayed. Ended up being admitted, it was actually mixed up by the grower, and the crop was Baxter. It was never Gregory. Okay, so just be careful. Like pub talk's great, but make sure it fits with your experience and what you're expecting as well. These things unfortunately happen. Here are least rough pub types. We won't go into this one. This is the one that's caused a problem in Naparoo. If you've got Naparoo, AGT sent specific information out to all registered Naparoo growers. That's fine. This is the exotic introduction into South Australia last year, and yeah, certainly they're worried about it down there. They got it in revenue, but it certainly was widely spread through South Australia after that. And this is the expected rating, so some of these are changing, some of our varieties are in here. I guess Spitfire, but Spitfire is already moderately susceptible. Limited information at the moment, I'll get better data this year, but just one to keep an eye on. The thing I want to make around leaf rust, you probably only pathologists who pick up on these things, but if you do hear about these new pathotypes, keep it in perspective, okay? Very exciting for pathologists, we love it. Keeps us in a job, but keep it in perspective. So in terms of leaf rust, we've got moderately susceptible varieties out there to existing pathotypes. So Spitfire's already moderately susceptible. They just had to spray their Spitfire for leaf rust. It's there. Okay, so just because you hear a new pathotype doesn't mean it's actually going to cause you an issue. But just monitor and be aware. You happy to go on the next one? Let's keep going. Okay, yellow spot. So Chris brought in some samples. Quite often see it early, this seedling stage. Key point I want to make here, and we'll go into it, is we're talking about a necrotroph. It's not a biotroph. So a necrotroph kills the cells first, and then it feeds off the dead cells. And it kills it by producing a toxin, which is what gives the yellowing. The yellowing is the toxin production by the fungus, killing cells in advance of where it's going to feed next. Okay, and that's the key point. If you don't want it, don't say wheat on wheat, or SPC, it can survive. So generally, I read up on this last night, generally 80 to 90% of the spores will be released in the first year after, but that depends on rainfall. So it needs rainfall, moisture for the spores to mature on the stubble. If you get dry years, you can actually delay that maturity and you can get a significant amount into the second year. Sorry?
Is it the same with barley?
Yeah, okay, so this will come at the end, but exactly the same. Different species, but everything I say about yellow spot is exactly the same with the net blotches, just a different species. So barley is a barley on barley issue. So it's the barley stubble that hosts the net blotches. But it's still producing the toxin in advance of where they're both necrotrophs, both producing toxin in advance of where.
Barley on barley seems to handle a bit better than wheat on wheat, would you say?
Depends on there is varietal resistance, so it really depends. Yeah. Not as bad, yeah. But the issue with some of the barley too is just physiological spotting as well. But we'll cover that.
So one doesn't affect the other?
No. You're going to hear that too, croppies. No, they are crop specific, but what I think is happening with net blotch is a lot of the net blotch is misdiagnosed. The yellow spot fungus doesn't know whether it's landing on a wheat leaf or a barley leaf until it infects.
Yep. And
in the process of trying to infect, the resistance of the barley to the yellow spot fungus, you still get production of small brown spots, which I think quite often are getting called net blotch, spot form net blotch. But we'll go into that, yeah. Okay, again, probably don't burn down your fences, but yeah, late autumn burn. So in terms of yellow spot, which is a stubble-borne pathogen, if you burn that stubble off, you get rid of your inoculum source for yellow spot, okay? Not as effective for crown. You still get rid of crown rot inoculum by burning, but it can survive in the crown as well below ground. Which I know inter-row sowing probably hasn't taken off as much, but burning and inter-row sowing, getting away from the old crowns, can be quite effective for crown rot too.
Steve, just on that, so what about a burn and a cultivate? Does that spread it, or does that make it worse?
For crown rot?
Yeah, for crown rot.
Yeah, for crown rot, well, it really depends. There's no clear, never a clear answer. Really depends. So what you'll find as a general rule, you get reduced survival in the crown than above ground because it's in the soil, you've got moisture, you've got more microbial activity. And if you look at, as we head further north, we have had decent summer rain, like up around Tamworth, Moree. Survival in the crown is way down this year, but there's significant survival. So some of the numbers, like 10%, so on the same stem, 10% recovery from the crown, 70% in the above ground stubble. So yeah, in terms of spreading around, I would leave it alone generally. So what you do is you just increase contact between the emerging seedling. Yeah. So the main infection point, one of the main infection points for crown rot is the coleoptile, the first structure that pushes to the top. There's no resistance at all in coleoptiles to infection by the crown rot fungus. So if you can try to limit that contact, then yeah, you limit the number of plants that get infected. And then the bump-on benefit is the fewer plants that get infected, the less stubble that's got the fungus in it, the less you're dealing with through the cycle. So yeah, stubble's an issue for sowing, you know, with some of that sowing thing, but in terms of just crown rot for the disease site, it's not the stubble as such that's the problem, it's the amount of the crown rot fungus in it that's the issue. Okay, so if we can limit that and keep that, then yeah, we're fine. But the problem we always run into is that decomposition process. And we run into dry years, and what we're really chasing a lot at the moment is 2010. You can see that through Predictor B, which is a DNA test, and the stuff we do. We're really seeing a bump up in crown rot levels after 2010. Big year, heaps of stubble went back in. That moisture is very conducive to infection by the crown rot fungus. You don't see expression of whiteheads. So we've increased our infection levels and our stubble loads from that. and then we're chasing that through the cycle. So, and then you go to drying properties, you don't get the breakdown, you don't see it decline as quick. That makes sense? Yeah.